Welcome, everybody. This is not our anniversary of our speaker series. It's sort of the anniversary of the pandemic. So this is a totally wonderful talk that we're having. Northampton Neighbors is sponsoring this. We went to Zoom in May. We have over 900 members, and our goal is to help people age in place. And one of those critical things are to keep us occupied in our mental health good. And these talks, as long as you have the solution, Marshall, these talks are going to keep us alive and going far. Marshall will talk for 35 minutes, and then he'll take questions. You can, if you would, try to hold off on your questions because he may answer them as he speaks, but otherwise put them in the chat button, or you'll have an opportunity we'll unmute you if you really want to ask in person or clarify something. A couple of technical things. First of all, Nina is here and she's our tech person. She's going to record this. She's also available when I get stuck or if Marshall should get stuck. In the upper left-hand corner, we have Otter, which is a great transcription service. So if you want to read it, you may do that. We're going to mute everybody, or you're all muted now so that we don't have any background noise and we can hear Marshall well. Just a little information. Our next two speakers are on 3/26 is Jeff Zessinger, and he's going to talk about what the heck is palliative care. And then on 4/9, April already, we'll have Howie Bryant, and he's going to talk about race and sports. He's just published a book on it, and so it should be interesting. So Marshall, I'm going to introduce you. Marshall grew up right in Northampton. He went to Bates College for his undergraduate degree. Then he did work at Harvard, MIT, MGH, studying nanoparticles targeted for therapy. And he's now working with the virology program at Harvard since 2015. And he's working on vaccine responses in the context of immune tolerance. We're really delighted and happy to welcome Marshall, and you can take it away and let us know everything about these vaccines and why we want them. Go for it, Marshall, all yours.
Thank you very much. I'm going to start by sharing my screen. There we go. Thank you very much for having me. I have an awful lot to get through today. So I'm going to go fairly quickly. I did want to start off with some disclosures. I have no competing or conflicting interests. I have no financial interests or financial disclosures to make on this topic. I'm doing this for two reasons. One, they're pretty standard in my field. And also, given the amount of misinformation and outright disinformation that's out there right now, both about COVID and about vaccines in general, I feel that steps like this are not only helpful, but necessary. Slightly less formal disclosures, things that I know a little bit about include viruses, vaccines, and not medicine. I am not a medical doctor. If you have questions about vaccinations or vaccines or immunology or virology or vaccinology, by all means ask me if you have questions about your particular medical care. I'm useless. I should mention that anybody who isn't actually staring at your specific medical history at that time is also useless, so I don't feel too bad about that. But if I get questions about specifically what any particular individual should do, I think those should be best addressed by your PCP. So my aims for this talk were originally to talk about COVID and the COVID vaccines. As I started thinking about how to put this together, I realized that in order to explain that, I really needed to explain normal vaccines. And as I started pulling together the actual slides for this, I realized that in order to explain COVID, I needed to explain about half of undergraduate molecular biology. And to explain how COVID works, I needed to explain a decent amount of virology. To explain vaccines, I need to explain immunology. And to explain the COVID vaccines, I need to explain what traditional vaccines themselves are like and how they're different from what we might have been having over the last couple of decades. So what turned into some very simple aims turned into a much more prolonged overview. My goal is to get through all of this in about 35 minutes. And to do that, I will be engaging in criminal oversimplification. Almost nothing that I say here will be uncontradicted in some aspect of human biology or greater biology at large. So if you are an expert in any of these particular fields, and this feels like nails on a chalkboard, just hold it until the end and then you can berate me then. I also do want to mention the SARS-CoV-2 virus is responsible for causing the disease known as COVID-19. But for the entire purpose of this talk, in order to save precious seconds, I'm just going to refer to it as COVID from beginning to end. So we're going to jump right in here with some molecular biology. And the reason that we're specifically going through that is because COVID is essentially just RNA, proteins, and lipids. The vaccines that we're going to be talking about later are either proteins or RNA, DNA, and lipids. And so therefore, running through these very quickly is not actually a waste of our time. There's going to be a very small number of slides in which I say this is actually worth remembering, because for the most part here, my goal is not to provide specific information, but rather a framework with which you'll be able to approach more news as you read about it in the future. If you hear about a particular protein, or if you hear about a way that a virus infects a cell, having made sense of this, you'll be better prepared to interpret those. But very little here is actually worth remembering. This is one of those few slides. This is what's referred to as the central dogma of cell biology. We shouldn't have dogmas in science, but still. DNA leads to RNA, leads to protein. That's the central dogma. Now, your DNA is your genome. You get half of it from your mother, half of it from your father. It's essentially a huge library that sits inside the nucleus of your cell. And it's specifically a non-lending library in that it never leaves. Parts of it don't go to the rest of the cell. In order for it to be read by the cell, it's actually unwound, and then a photocopy is made, which is RNA. Your DNA, you only have one copy of your genome per cell. And if it were translated into letters, A, T, C, G, it's about 3 billion letters long. It codes for roughly 3,000 different proteins, and you can think of each gene as being a blueprint for a protein. Your genome stays the same, and it's the same in all of your cells. All of your cells have exactly the same genome. What makes a toenail cell different from an eyeball cell, from a spleen cell, is what parts of your genome that those cells are reading. And the reading of it is your RNA. So if your genome, your DNA, is a huge non-lending library of 30,000 blueprints, each RNA, in this case messenger RNA, is a single flimsy photocopy. You have millions of them in each cell at any time. They're always changing. They're only a few hundred letters long each, typically. And these are far more mutable than a genome might be. So if your DNA is a library of 30,000 or so blueprints, and if each mRNA is a photocopy of a single blueprint, then the question is, blueprint of what? The answer here, obviously, is proteins. mRNA and DNA are very, very chemically similar. They're both made out of nucleotides, and their purpose is information storage. Proteins are completely different. For one thing, they're made out of amino acids. You might have heard about them in some like nutritional context or things like that. Tryptophan, if you eat too much turkey, that's an amino acid. And the purpose of proteins is a little bit more diverse than that of DNA and RNA. Proteins are incredibly complex molecular machines. They cut things, they chop things, they splice things, they open them, they close them, they unwind them. Anything that needs to move at the molecular level is probably being moved by a protein. Last but not least is lipids. This is my very artistic rendering of a lipid there on the left. They come in two parts, essentially. The head of the lipid is hydrophilic. The tail is hydrophobic. And that means that if you throw these into a big pot of water, the tails are going to try to get away from the water, and the heads are going to try to get closer. So if you have enough, they'll form what's known as a little micelle like this, where all the tails are sequestered from the water. If you have enough of them, they form what's known as a bilipid layer, where you can have water outside the bubble and inside the bubble. And that's what a cell is. The skin of a cell is made of lipids. And these lipids can all move back and forth next to each other. It's called a fluid mosaic model. They're all bouncing around all the time. They're not held firmly in place. But this is what the skin of a cell looks like. And with that, we've pretty much reviewed most of undergraduate molecular biology. I did think of a few ways in which I could tie this back to practical experience. What else forms bubbles in water besides oil? Soap. The way that soap cuts oil when you're doing your dishes is that soap has a hydrophobic tail somewhat similar to oil. So it can insert that hydrophobic tail into the oil, break it apart, and lift it away. That's also why washing your hands can kill coronavirus. Coronavirus has a lipid bilayer on the outside, just like your cells. And when you wash it with soap, the soap intercalates into the membranes, disrupts the membranes, and essentially pops COVID like a little drop of oil is popped by soap. So like I said, we just breezed through all of molecular biology in a couple of minutes, and now we're going to move into virology. What is a virus? How does it work? And so I'm going to start that by talking about cells. Also, I'm just reminded that if you have the panel of faces off to the right, as I do, you're going to lose a little bit of my next couple of slides. So if you want to move that now is a good time. Cells are tremendously complex. Here's a little artist's depiction of a cell over there on the left. One of the most important things about them is that they have metabolism. They eat and breathe and poop. They have the capacity for self-renewal and, like I said, tremendous complexity. The analogy that I'm going to be using for most of this talk is that they are a city block-sized factory capable of making 30,000 different things. Each of those 30,000 different things are parts of the machines inside the factory, such that the factory can infinitely replace its own parts as they wear out, to the extent that it can even make an entire second factory, which is what happens every time a cell divides. Viruses have none of these things. They are not complex. They have no capacity for self-renewal. They are parasites that cannot replicate themselves. And they have no metabolism whatsoever. They do not eat or breathe or poop. Roughly speaking, because there's tremendous variability in the viral world, a virus is roughly the same size as the period at the end of this nope compared to the cell. Part of the reason they can be that small is because they require none of the complexity that a cell does, because they steal that from the cell. So the cell needs to use a nucleus and a nucleolus, and here's some mitochondria. You've got an endoplasmic reticulum, you've got a Golgi apparatus, you have centrioles and centrosomes and nucleosomes and proteasomes and all these other things that are required to sustain life. The virus doesn't need any of those things because it just gets your cell and then uses yours to make more of itself. So if a cell is a city block size factory of near infinite complexity, a virus is a manila envelope containing a single blueprint for more manila envelopes. And then it gets inside the cell and tells the cell to make more manila envelopes. It's basically like biological hate mail. I've included some pictures because a picture is worth 1,000 words. We have HIV at the top here, and we have some influenza here at the bottom. So this is false color, but the blue is an infected cell, and the green are the little HIV particles that are popping out. Specifically in the case of COVID, and I got this picture from the New York Times, the virus is quite simple. You have a couple of proteins on the outside, which are known as the spike proteins. These are important, and we'll be coming back to them. They help the virus get inside the cell, and they're what your immune system likes to target when it can. It also then has a lipid bilayer, and this actually, because COVID is sneaky, it's derived from the host cell's plasma membrane. So when the virus leaves your cell, it rips off a piece of skin with it, of cell skin, wraps itself up with it, and then uses that to hide from the immune system. And then inside is the viral genome. Now our genome is made of DNA, and it's immutable. It sits inside the nucleus and never goes anywhere. The viral genomes are RNA. So they're coming in halfway through that central dogma and messing everything up. So here's how infection works in a very simplified level. This is a normal human cell doing normal human cell things. This is outside the cell. This is inside the cell. Here we have a nucleus with human DNA inside it. mRNA is being transcribed from that and then translated into normal human proteins. This is what happens when the cell just needs to sustain itself normally over time. This is what happened when COVID gets in. The first thing that it does is it binds to receptors on the outside of the cell. You might have heard of the ACE2 receptor being mentioned in this context. Once it's inside, it pops itself open, decapsulates its genome, and then sends its genome off into the cell. And like I said earlier, its genome isn't DNA that sits in the nucleus like ours does. Its genome is RNA, and it pretends to be human RNA. So the human cell starts to copy this and to turn it into more COVID proteins, and then it stops sustaining itself. So part of the reasons that your cells die when they become infected with viruses is because they stop doing self-care. They start caring for making more viruses. Now they aren't replacing their own parts as they wear out, and the cell goes downhill. There's lots of other reasons, but that's a main one. These viral components, the viral proteins that are being made by the confused cell, they self-assemble on their own. And then they stick one of the new genomes inside themselves, and then they pop out and they steal some of the cell membrane with them, and they restart that whole process. So the technical term for a virus is an obligate intracellular parasite, which is to say something that has to get inside a cell in order to make more of itself, because like I said, it's basically biological hate mail. So that was all of virology in about 5 minutes as well. We're cruising right along, and now we get into immunology, which is notoriously, hilariously, ridiculously complicated. And I don't know if I've really condensed it well, but we're gonna have to go through it anyway. Specifically, the human immune response is roughly broken down into two components, the innate immune system and the adaptive. The innate immune system is pre-programmed from birth. If you are a human, then you automatically have a paid subscription to the magazine known as Innate Immunity. And it's for very general things like inflammation. Your body doesn't need to know exactly what's wrong in order to trigger inflammation. More than 2,000 years ago, people were already recording the four hallmarks of inflammation as they're known now, which is pain, redness, swelling, and warmth. But the innate immune system can't adapt. It can't learn over time. That's what the adaptive immune system can do. Now, it's utterly useless at birth. It needs to be taught everything because it's never encountered any antigens before, but it can learn, adapt, and remember. And the remembering part is the important part. So now we're going to go through a hypothetical example of COVID infecting some cells and the immune system responding. We're down in some poor individual's lung right now. This is the airspace. This is a ring of cells, and this is your bloodstream, but right below that. This is COVID again. Not quite to scale because it's hard to depict here, but I've made some little COVIDs that will serve our purpose for now. Starts by getting inside a cell, pops open, infects the cell, and tells the cell to start making more COVID proteins. And I've made a bunch of little spike proteins here because those are proteins that we're interested in. As the cell remains infected for more time, you start to get the self-assembly of new viruses. They leave the cell, infect the cells nearby, and the process starts again. Now, as that cell is dying and as other cells are infected, you get inflammation. And I've put my very technical inflammation little red bubble there. The cells of the immune system, by and large, live in places like lymph nodes and spleens, but there are sentinel cells that live in all of your tissues all of the time, always looking out for danger. Neutrophils are one kind of example. And neutrophils, if they're flying through your bloodstream and they detect inflammation nearby, will immediately put on the brakes, crawl through your bloodstream, and go into the infected tissue. And their job is to hunt for infections. In particular, they really love bacteria. And when I say hunt for infections, I really do mean hunt for infections. In this short and very technical video, you have very small bacteria here in black. You have red blood cells here as more circles. And this guy here is a neutrophil that is trying to eat the bacteria. So that's neutrophils. Macrophages are also in your tissues at all times, and they're another great immune system cell. And their job is to basically garbage truck. Dead cells, dead viruses, dead bacteria, live viruses, live bacteria. It eats all of them. And as it does it, it does what's probably the most important part of the whole lecture that I'll be giving today, which is antigen presentation. So here's one of the spike proteins. It's gobbling it up. It then breaks it down and then puts the pieces that it found on its own cell surface. Now I'm going to highlight this in a whole separate slide because, like I said, this is the most important thing that I'll be talking about today. Antigens are worth remembering. An antigen is anything that provokes an immune response. Unfortunately, pollen is an antigen, even though it shouldn't be. But for the most part, this is the important parts of dead viruses and dead bacteria. So when it breaks down this spike protein here, it breaks it into discrete subcomponents, small parts of protein, and then it mounts those on its exterior. And I tried to come up with a better comparison to make here, but honestly, the best comparison I can think of is it's roughly equivalent to mounting the decapitated heads of a defeated foe on spikes. The goal is to run around the neighborhood showing everybody exactly what the danger is, what it looks like, and how to fight it. And that is antigen presentation, and it's the fundamental basis of the adaptive immune response, which we get to move into now. So the adaptive immune system, for the most part, starts in lymph nodes, which is why when you get an infection, it gets all swollen. You get three main cell types, though there are many others that participate in this process. You have your antigen presenting cells, which we covered before. Their job is to provide the antigen, which is the small pieces of the defeated bacteria or virus, to the B and T cells. B and T sound very familiar, so I came up with some helpful ways to remember it. The T cells are like teachers. Their job is to teach your B cells, and the B cells are like blacksmiths because they're the ones that actually make the antibodies, which are so critical to how vaccines function. Because this is immunology, the T cells wear multiple hats, and in addition to constructively teaching your B cells, they also terminate and murder all of your infected cells. So the T cells wear more than one hat. Here we are back at our infection. Now that the antigen presenting cells have carried the viral antigens to the lymph node and educated those cells now leave and enter your bloodstream. When they encounter inflammation, they're going to home in on it just like the innate cells did, but now they've been trained to recognize that spike protein because of what happened in the lymph node. And as soon as this T cell recognizes that this cell has been infected by COVID, it goes into terminator mode and kills it. So this is the way that your adaptive immune system has learned to clear the virus, which is inside your cells. And it can survey almost any cell in the immune system. The question is, how do you get rid of the virus that isn't currently in your cells? If it's floating around in your blood or lymph or tears or something like that. And the answer there is B cells. Now, these guys can be sitting in your spleen miles away, relatively, immunologically speaking, on the other side of your body, and they secrete antibodies, which are small proteins, into your bloodstream. And now antibodies do a lot of different things, but the simplest thing that they do is to bind to this virus and prevent it from further infecting cells. Now, if that virus is floating around in your bloodstream, but it's unable to infect anything, it's essentially dead. And it will get cleared out by your immune system over time, in part by things like macrophages, which will gobble it up. So between the B and the T cells, that's how the adaptive immune system can account for all the virus in your body, whether it's inside your cells or outside your cells. The way that actually works is different whether you've been infected by this virus once before or not. A primary infection is one that you've never encountered before. So this would be when you send your toddler off to preschool for the first time and they come back and they get sick once per week for 6 months. It's because they've never encountered any of those immunogens before. On day one, the first thing that happens is this antigen presentation happens between an APC, antigen presenting cell, and a B cell. As soon as that B cell gets activated on day one, it begins to proliferate, and the daughter cells split into, roughly speaking, two groups. One group's job is to make antibodies. These cells live fast and die young. As soon as they're activated, they produce antibody for a short period of time, and then they die. This other cohort, their job is to improve the antibodies over time. This is called affinity maturation or somatic hypermutation. The words are important, but you might come across them on your own. This process takes roughly 2 to 3 weeks, which you might recognize from some of the vaccine schedules. Periodically throughout this process, some of the B cells are kicked out into this other group so that rather than waiting 3 weeks to make good antibodies, that process can gradually occur over the course of this 3-week infection period, because this is a critical time. If you have something like Ebola, you don't have 2 months to wait for a good, strong immune response. Whether or not you survive can be determined to a great extent by how effective your initial immune response is. At the end of this process comes the fun part, the truly adaptive part, which is some of these B cells turn into memory B cells, whose job is literally to sit around and do nothing for decades. Their job is only to wait around in case you are ever infected by that same bug again the second time. If you encounter that virus again for a second time, now that's a secondary immune response. To extend the analogy from earlier, now we're talking more about a kindergarten teacher who's been doing it for 30 years. Their immune system has seen everything under the sun. They no longer get sick because for them, everything is a secondary infection. And what happens here is roughly similar. On day one, antigen is presented to both naive B cells and also the memory B cells that remember the antigen from the previous infection. Both of those B cells begin to start making improved antibodies. They're both taking their nice, sweet time. It's going to take 3 weeks for that process to happen. But while that's happening, the activation of memory B cells is slightly more dramatic. They don't wait around. On day one, they immediately start proliferating, producing tons of high affinity, strong targeted antibodies, and they dump this into your blood. So you don't have to wait 3 weeks again. This way, for a secondary immune response, you get the benefits of the first infection pretty much on day 2. And this is the goal of vaccination, to get you to the place where you can have a secondary immune response to a primary infection, which neatly brings us all the way to vaccinology. We've now covered all of molecular biology, a bit of virology, and most of immunology, if I'm being honest. So now I'm going to talk briefly about some of the traditional vaccines that we might have grown up with, and then end with a little bit about the COVID vaccines. So the goal of vaccination is to reduce the severity of infectious disease. It's not to prevent reinfection. And we'll probably cover that again when we start going through questions later. The mechanism is the same regardless of any vaccine, which is to provide the immune system with antigens to study. If you're trying to immunize against polio, you provide polio antigens. If you're trying to immunize against tetanus, you provide tetanus antigen. The method for that changes with technology, and it always has. The outcome, though, is that a fake infection without disease will prevent disease when the infection is real. That's the goal. And I've reiterated this in the context of my earlier slide because it's this antigen presentation step that's so important and is the basis for these vaccines. And I've broken the different vaccine modalities up here at the bottom to go through one by one. I should mention the history of vaccinology is not my area of expertise. I've come across this specific reference several times in my career, but I've never actually checked it carefully, so don't hold me to this. But roughly 1,000 years ago is the first evidence I can find of vaccines being used, and they were used in China against smallpox. And they were essentially the simplest type of vaccines, which is let's give people actual smallpox in small doses so that they won't get as bad a case of smallpox as they would normally. So here they would actually take the pustules from somebody who survived smallpox, never from someone who died of it, and use those to infect another individual who, if they survived, they would get the pus from one of their pustules and use it to infect another person. And over time, they ended up with slightly less deadly forms of smallpox. As we go through these different types of vaccines, I've put these little bars here to indicate the decreasing risk of infection, unfortunately, means a decreasing strength of the immune response. The immune system responds best to danger. And if you're using less and less dangerous viruses, you're going to be stimulating less and less strong immune responses. Unfortunately, you also have, or rather, fortunately, you have less and less risk of disease with improving vaccine modalities. The oldest vaccine, like this one, you are actually infecting somebody with the agent, which will cause the disease, and you merely hope that it's less severe. The slightly better version of that, some people may have encountered the comparison, skin as fair as a milkmaid. If you were born in Europe or Asia or Africa, for most of recorded history, you got smallpox at some point and had horrible facial scarring. Egyptian mummies, for the most part, all have smallpox scars that have been so well preserved that we're able to see those things. One of the few exceptions, generally speaking, were milkmaids who had less substantial facial scarring, in part because they got cowpox from milking the cows. So this is an example of an inadvertent vaccination. And here, they're not being infected with smallpox to prevent smallpox, they're being infected with cowpox to prevent smallpox. So it's a step in the right direction. You get a weaker immune response, you get less risk of infection and less risk of disease. Moving into more modern times, I've highlighted both attenuated and inactivated here because polio, I think, is such a good example of what vaccines are capable of doing, and also why improving vaccines continues to be important. The original smallpox vaccine was an attenuated smallpox. Attenuated in this case means a weakened version of the pathogen itself. So if you grow a virus in unideal circumstances, if you grow it in cells from a different species at a different temperature than the human body, it will adapt over time so that it can replicate well in those conditions. And then when you stick that into a person, it doesn't grow well at all. But it still provides the antigens that you want. So the original polio vaccine, the oral polio vaccine, was actually a very slightly mutated version of the original polio virus. And in very rare cases, it could revert to cause actual polio. By and large, this didn't happen. It happened incredibly infrequently, about 3 people per million. By comparison, about 5,000 people of those would go on to become paralyzed with polio if they hadn't been immunized. So in the end, it was still perfectly fine. But we still have vaccines that are available today that are attenuated, and their risks are far lower than the old polio. The reason I've highlighted both of these things is that the newer polio vaccine, the intramuscular one, the shot, most of our flu, our Hep A, rabies, things like that, are inactivated. So here they're taking the actual pathogen, the actual polio virus or the actual flu virus, and they're chemically killing it. So it's completely dead. And then they're injecting that as a way to provide antigens to the immune system. The only reason that I've included this infection bar all the way into inactivated is to highlight the hypothetical possibility that if somewhere during the manufacturing process something went horribly wrong, it is physically possible for an infectious particle to end up in a person. To my knowledge, it has absolutely never happened, but I'm including that because I think it's important to distinguish between that, which is hypothetically possible, and actual infections arising from subunit or nucleic acid vaccines, which are literally impossible. So the way a subunit vaccine works is you take some of the pathogen, you break it apart, you purify only the parts that are the antigens you want, and you put those into people. So the actual pathogen is gone. There is no more virus in that preparation whatsoever, and there's no way it can infect you. It's just a small part of that virus. Now, the original Hep B vaccine was actually, like, the source of the antigens came from the blood of infected individuals. A couple of years after that, they found a better way to do it, which is to take the genome of the hepatitis V virus, put it into yeast, let the yeast make the proteins they want, collect those, and inject them into people. And so that's how most subunit vaccines are made these days, which is you put the genome of these bugs into yeast or E. coli, which then serve as little tiny bioreactors and make your protein of interest for you. And then those are injected into people. And there's no possible way that you can be infected with those diseases because the actual pathogen isn't there. It's only a tiny piece of them. Now, as we proceed across this axis here to the right, as these get safer and safer and safer, they also get weaker and weaker and weaker. Your immune system isn't under attack. You're not getting as much cell death. You're not going to get as much antigen presentation. So it's roughly around the subunit vaccine level that you start to see adjuvants. And adjuvants are small chemicals that are used to worry the immune response, to freak it out, to make it think that this infection is more severe than it is, because there actually is no infection. A lot of anti-vax concern and anxiety comes from the use of adjuvants. And I'm, of course, happy to go into detail on that later. The DNA and the RNA vaccines are newer. And so this is harkening back to what I said earlier about how vaccine methods change with technology. For a subunit vaccine, you take the genome of the pathogen of interest, you put it into yeast, you collect the antigens using purification in a lab, and you put those into the vaccine. With the DNA or RNA vaccines, you cut out the middleman. You take the genome, you put that into a needle, you put that into somebody's arm, and that person's arm makes the vaccines right there on site, instead of having to go through all the trouble of doing purification from yeast. And so some of the new Ebola vaccines and some of the new COVID vaccines use this modality. So we're actually very nearly the end. All we have left are the COVID vaccines. There's about 40 or more that are in late phase trials at the moment. I'm only going to briefly review 4. And they divide up pretty well between the Pfizer/BioNTech and Moderna, both of which are mRNA inside lipids. We've covered both mRNA and lipids today, so you should have some idea how they work. Then there's the J&J and Oxford AstraZeneca, which are DNA inside a hollow virus. Covering both of these very briefly, here on the left, we have a reiteration of my earlier slides, where COVID gets into one of these cells, makes it sick, makes more COVID, explodes the cell, goes on, creates this whole immune response, followed by antigen presentation, which is the end goal. Over here, we have the mRNA inside lipid vaccines, Pfizer and Moderna. What they did here is they essentially recreated the lipids that surround the virus, but reproduced them in a lab and simplified them. And then they put inside that the COVID genome. But instead of the whole COVID genome, because if they did that, you would start making COVID, they put only a small fraction of it. They put only the portion of the COVID genome that codes for the spike protein, which is what they're trying to present to the immune system. So they take that small portion of the COVID genome, they wrap it in a lipid bubble, and they throw it into your arm. The lipids from the bubble bind with the lipids in your cell, and then the next thing you know, that little piece of genetic material is inside the cell, telling it to start making the COVID spike protein. It does so, and then boom, you've got the antigen presentation that would have originally required a full infection to cause. The J&J and AstraZeneca is essentially the same thing, just uses a different vehicle to get inside the body. So instead of using a lipid nanoparticle, they use an empty virus known as adenovirus. Johnson & Johnson, I believe, uses adenovirus 26, and the Oxford, I believe, uses actually a chimpanzee-derived adenovirus. And what they do there is they essentially take the adenovirus genome out, because if you have an adenovirus genome inside an adenovirus, you're going to get adenoviruses out of the cells. And adenoviruses cause common cold-like symptoms, but you don't want that if you can avoid it. So they take essentially the same part of the COVID genome, just the part that makes the spike protein, and they throw that inside an empty adenovirus. And the reason they do that is because adenoviruses are experts at getting inside human cells. They've been doing it for millions of years. So here they capitalize on that experience. They throw the portion of the COVID genome that makes these spike proteins inside an empty adenovirus. They put that into your arm. It gets inside your cells, starts making more spike proteins, and you end up with antigen presentation at the end, just like they had hoped. Efficacy. I always forget how efficacy is calculated, so I threw this up because I thought people might have questions. I've highlighted the Moderna trial up here and the Pfizer trial down here. I'm happy to go through this in as much detail as people like, but I highlighted the part that I thought was the most important. So they enrolled about 15,000 people to give the placebo to, and about 15,000 people to give the vaccine to. Some of these didn't get both, or they dropped out of the study. The actual number changed a little bit before the end of the actual trial, but they stopped at the trial when they had found enough infections to report accurately whether these things are protective or not. And once you've done all the statistics, it turns out to be about this many. So they got about 185 people who had been given the placebo who got COVID. In that same time period, only 11 of the people who had gotten the vaccine got COVID. And that's all well and good. But I've highlighted in black the part that I think is the most important, which is the severe infection, the people who were hospitalized. Of the 15,000 people who got the Moderna vaccine, none of them were hospitalized for COVID. And that, I think, is the most important, because COVID is not going away anytime soon. To hope that you don't get COVID is to hope that you don't get the flu. If you wait long enough, it's probably going to happen at some point or another. But if you have gotten these vaccines, your odds of going to the hospital are, and we can say this with some statistical certainty, slim to none. They had one COVID death during this cohort and none in the vaccine. Down here, the Pfizer trial is roughly similar, a slightly less protection from the severe COVID, but when you magnify this across the actual pool of the number of individuals who've gotten this, they're actually quite similar. For the calculation of efficacy, I put that down here in case anybody had questions about that. In a hypothetical case where the Pfizer vaccine had zero efficacy, we would expect to see 162 people who got COVID in their placebo. And if it does nothing at all, you'd expect 162 people in the vaccine group. A hypothetical vaccine that had 100% efficacy, we would see that drop to zero. So efficacy, a 95% efficacy doesn't mean that you have a 95% less chance of getting infected. The stats are slightly different from that, but it's essentially the same thing. And I've included the severe reactions component up here because I think it's easy when looking at numbers like this to forget that they're taking place in the context of people's lives. These aren't numbers, these are people, and they have other things going on in their life. And in the roughly 30,000 people in the Moderna trial, 4 people died. One of them died of COVID, and a number of other people died from other things, including suicide, heart attack, bowel perforation. These aren't rats living in a tube somewhere in a building. These are real people, and it reflects the real complexity that's going on in everyone's life. Which is why I see numbers like 1 here versus none here, and I'm not necessarily going to come out then and say that the Pfizer vaccine is worse than the Moderna vaccine. You need larger numbers to demonstrate that sort of thing. So having run you guys through a whole lot of content in a very short period of time, I'm happy to take questions if we run out of time, or if you have questions that you don't want to ask there, I'd invite you to email me directly. And I've also put up the Moderna and Pfizer vaccine list, the ingredient list, because I think that this is particularly interesting, particularly for people who might have concerns about components that used to be present in vaccines. I think it's reassuring to see that it's basically now a couple of lipids and a bunch of food grade reagents. Thank you very much for your time and attention. And I'm going to stop sharing my screen now.
Nina, can I get highlighted again, please? Thank you. We can't clap. but that was a spectacular presentation. Thank you very much. There's some, there's some clapping, Zoom clapping. Just before we go on to questions, and please, I see some are coming in on chat. Many so far have said, wonderful presentation, but we'll get more questions, I'm sure. But I just have to make a short ask for Northampton neighbors. We are somewhat unique because we don't charge any membership fees or any fees for any of our services. Our services have taken a little hiatus over the pandemic, and that's one of the reasons the speaker series is so important. We also do calls to people who would like weekly check-in calls. But we do survive on donations and grant writing. And so if anyone wants to give a donation, no matter how small, it would be wonderful for us to have. You just have to go to northamptonneighbors.org and hit the donate button, and you can pay for any credit card or PayPal, or you can send in a check. We'd be most appreciative. It's not necessary, but it's wanted desperately. Not desperately, it's wanted. So let's go on to the questions, and I'm going to try to look at them. Nina's going to unmute people who have questions, and so if you can go to the participant, there's a raised hand you can put up virtually, and that'll help Nina move you and unmute you. Is that correct, Nina?
No, actually, people can unmute themselves if they have a question, or they can raise their digital hand.
Okay. So far, one of the questions is it safe for a very sick long hauler to get vaccinated?
Depends on your medical history. Hard to say. Very sick can mean a lot of different things. And so it's hard for me to answer that without knowing specifically what you're referring to. My guess is yes, but don't hold me to that, and definitely ask your PCP.
That is a medical question.
Definitely.
If you know what type of vaccine is in Shingrix.
Yes, Shingrix is very interesting. That's a subunit vaccine, I believe. So I had said that the goal of vaccination is to reduce the severity associated with viral infections, or in this case, bacterial infections as well. Some people may or may not be aware that shingles is actually just chickenpox round 2. So when you get chickenpox as a kid, you get itchy bumps, they go away. It actually sits inside your nerves for about 50 years and then comes back out later to make your life miserable. Part of the reason that shingles hurts so much is because it's actually coming out of your nerves itself. That's why some people tend to get it in radial patterns along their back. It's following the nerves that radiate out of the spinal column. Other places that you get it in your body, it's similar, might not be quite as anatomically exact, but it's coming out of your nerves decades after you last had chickenpox, usually when your immune system starts to get weakened. That can be if you got a transplant and you're on immunosuppressive therapy, or just with age, or HIV infection. Shingles in very, very young adults, 20, 30, is sometimes one of the things that they'll often check for is HIV infection or things like that. The Shingrix vaccine is designed to prevent zoster, which is shingles and things like that. But the fun thing is that hopefully in the next couple of decades, it's going to put itself out of business, because kids these days aren't going to chickenpox parties and getting chickenpox. They're getting a chickenpox vaccine, which means that when they finally get infected with chickenpox a year or two later in the normal course of their lives, it doesn't end up in their nerves, which means hopefully we have an entire generation of kids that are born now who will never have to get shingles later on. But the quick answer is subunit.
Okay. The questions are coming in now. What is the distinction, not being a medical person, but not if you can answer, between what is a severe versus a mild reaction?
A severe or mild reaction to immunization or to infection?
Yes, yeah, to the vaccine. The vaccine, what's the distinction if it causes a mild versus a severe?
Depends on which one you're talking about. I know that's not a very helpful answer, but these companies tend not to play very nicely together. And so they don't design their vaccine trials with identical endpoints. So when one trial is created by Moderna and their goal is to measure efficacy and safety and adverse responses, what they consider an adverse response small or adverse response large will be using different criteria than Pfizer uses or BioNTech or anything else. So I can't give you a simple definition and a simple breakdown of what those differences are. Generally speaking, when these trials are designed, they're done so with the previous experience of what you normally get out of a vaccine, which is to say some pain in your arm, some redness and some swelling are to be expected. Once you've got somebody who's got vomiting, diarrhea, and anaphylaxis, that's not normal and far more likely to be included in these severe outcomes. But there's no firm line between for every single vaccine as to what that distinction is.
Thank you. There's talk about vaccines that might cover multiple cold viruses and variations in the future. Do you think that's realistic?
Yes, multiple, yes, all, no. So for a couple of reasons, particularly for things like flu. So coronavirus can only mutate in one way. Its genome will slowly change over time. That's how these variants are emerging. Flu can do that, and it can do something else too, which is actually two different flu particles. A pig flu and a bird flu can infect a person and then literally mix their genomes together because its genome is 11 different pieces. So it can take 5 pieces from one flu and 6 pieces from another flu, stick them together and make a completely new flu particle that nobody has seen. And now it's 1918 again, and 2% of the world's population is in trouble. What we call the common cold is a couple of different types of viruses that all cause similar symptoms. I do think it's realistic to say that 50 to 100 years from now, we will have a vaccine that covers the majority of them, but we certainly aren't going to get anything in the next 3 or 4 years that will make the common cold go away.
Not in my lifetime anyway.
And I'd love to be wrong about that.
Okay. Do you know if you are immune after a vaccine? And how long is one immune after the vaccine?
Good question. Yes, you can know. For how long, that's harder. So as vaccines have been in commercial production for longer, years or decades, we have a better idea of what to expect from how long they'll last. I think it's pretty common knowledge that if it's been 30 years since your last tetanus shot and you step on a rusty nail, it's time to go get another tetanus shot. We know that the average half-life of that is about 5 to 10 years, which is to say 5 to 10 years later, most people will have waning immunity. The yellow fever vaccine, the best calculation people are able to do is that it lasts roughly 300 years. So like if you've had the yellow fever vaccine, you're probably good to go. For these, there's absolutely no way we can know because people haven't had them in their bodies for long. There's a lot of evidence that immunity to COVID does not last very long, not nearly as long as it does for some other things like smallpox, where if you get that once, you're good for life. So it's hard to say for COVID. My guess is it won't be great and that we'll need boosters over time, but with the variants, it's likely we'll need boosters anyway. For things that have been on the market for a long time, like hepatitis B vaccine, generally speaking, if it's been about 10 years since your last Hep B vaccine, what they'll do before giving you another one is they'll draw a little bit of blood and then do a test on it to see what your antibody levels are. And if you have lots of Hep B antibodies, they'll say, yeah, you're good to go. So for each of these vaccines, we do have tests that will tell you approximately how well protected you are. We don't really have them for COVID yet, and we also don't know quite how long they'll last for COVID.
Do you know, another question is, are there any studies that people could join that would be testing this over time? I
am sure that there are, particularly if you're in an area that's covered by a good healthcare system. Most of Massachusetts falls very neatly into that category. I can't tell you exactly how to go about finding them, but I'm guessing that there's an awfully big interest right now, particularly in the age groups for which COVID is most appropriate, which might fit a few people who happen to be in this presentation.
Is it too hard to explain? If the virus can't replicate, how does it mutate?
I would need
Is that going to take too long?
No, the person who asked that question, I would need to ask about the context of the first part of that question. What do they mean if the virus can't mutate? I mean, if the virus can't replicate?
Oh, on one of your slides, you said viruses can't replicate. They just use your cell to make more of their own.
I see. Yes. So viruses can't make more viruses. Viruses can get inside your cell and convince your cell to make more viruses. And it's during that process that they can mutate. In particular, the RNA viruses mutate much more rapidly than the DNA viruses because our bodies spell check our DNA very stringently. If your DNA changes, you get cancer. If your RNA changes, you make a misfolded protein for a couple of days until that RNA breaks down in the natural course of time. So we don't spell check our RNA very well, but we do spell check our DNA very well. So the RNA viruses take advantage of that, and they'll produce tons of crappy viruses with lots of mutations in them, most of which will be detrimental to the virus and some of which will improve it, which is why things like HIV and COVID mutate so much more rapidly than some of the other viruses.
Okay. This question says, I'm asking about reactions that might be considered severe, but a day of headache, this is reactions to the vaccination, a day of headache, body aches, etc. Why do some people get those and others don't? Sometimes you hear that it's better if you have more of a reaction because it means your immune system is working better. Is that true?
Yes, that's true. Also, we don't know. So it's true that a vigorous immune response is suggestive of a vigorous immune response, which is to say, if you feel sick, it's because your immune system is responding, which is a good indication that you're going to be able to make the long-term antibodies that you need to make. It's not a guarantee. It also doesn't mean that people who don't feel like crap aren't going to make good antibodies. Often they do. The COVID vaccines that are out on the market now protect the elderly slightly less well than they protect young people, because the older immune systems don't get fired up quite as easily as younger immune systems, which is also why younger people tend to have more significant side effects than older people. However, we're talking about very, very general trends over a huge number of people. And so an individual person's response, like if you get a headache or if you don't, that shouldn't inform how confident you feel about having received your own vaccination. As to why some people do and other people don't, there's a lot of research into that right now, but that gets into the complexity of human immunology. A lot of it has to do with genetics, a little bit of it has to do with environment, and we haven't sorted out a good enough model to make a predictive analysis based on somebody's genome. How well will they tolerate the vaccine? So there's a lot of unknowns there.
On that kind of idea, if you are a non-responder to the Hep B vaccine, does that mean potentially you won't respond as strongly to the vaccine, to any of these vaccines?
Probably not. There's a few conditions I can imagine which might mediate both of those. The individuals with common variable immune deficiency, CVID, their B cells don't do that crazy rapid proliferation on the memory B cell slide as well as they should. So if you are an individual with CVID or perhaps undiagnosed CVID, then the failure to respond to Hep B would also predict failure to respond to COVID. For an individual person without any specific medical background that's been documented, no way to predict.
Okay. This is maybe just a clarification. Did you say that 1.5% of the people who got a placebo had a severe reaction?
I believe it was 1.3, but yes. And that's actually something that was seen in, it's seen all the time in all vaccine trials. If you tell people that you're putting a vaccine in their arm, a small proportion of them are going to have vomiting and diarrhea. And if you tell people that you're putting water in their arm, they won't. And if you don't tell them which is which, people who got water are still going to have nausea and vomiting. Placebo is very cool, very weird. We don't understand it, but we know that it works. So the important statistic is never how many people who got the vaccine had a severe response, but how many more had a severe response than those who got the placebo. In this case, I believe the Moderna numbers were 1.5 versus 1.3.
Good answer, because placebo responses work in about 30% of all sicknesses, right?
It strongly depends, as from what little I know about placebo, on how it's administered. So for example, placebo acupuncture has a larger effect than placebo acupressure. If you tell people we're poking you with needles, it seems more invasive, more extreme. If you give people a red pill versus a green pill, same sort of thing. Our perception of the intervention strongly colors the effectiveness of placebo. And that is far outside my area of expertise, so I have no real numbers to cite on that. But to say that it governs 30% of infections is something I would believe. I would say it would be more like 100, and I just don't know how those numbers would actually be tested. Because how do you test a placebo? It's been a fun drama for a long time in the scientific community.
How quickly will viral mutations be addressed in booster shots?
Quickly. So one of the advantages of the mRNA vaccines is that you don't have to get some of the genome, put it into yeast, grow the yeast, kill the yeast, get the antigens out, purify the antigens, put them into a shot, and then give them to people. Here, you can just get your machine to produce a different mRNA than before. And so part of the reason that the mRNA vaccines hit the market fastest is because they're easiest to make, manufacture, and change, which means they're also going to be the easiest to mutate, manufacture, and change. The real question will be whether full vaccine trials are deemed necessary for them or not. The argument for that would be you're modulating somebody's immune system and autoimmunity is bad, so we should probably test this. The argument in favor of it is we've been doing that with the flu for decades. So that's going to be a fun ethical question for pay grades far above mine.
Okay. We're told that we're protected 2 weeks after the last vaccination. Do we start getting more protected daily by the shots, or does it by the vaccine, or does it take the full 2 weeks and then suddenly we're safer?
Biologically, it's more gradual. Statistically, it's all of a sudden, because when they were doing these trials, they didn't break down the trial into every single person who got it on day 14, and then every single person who got it on day 15, and then every single person who got it on day 16. They did people who got COVID before the second boost, or 4 weeks after the second boost, or maybe their trial was 3 weeks after the second boost, but they didn't test every day. So they have the statistics to tell you how protected you are at 2 weeks post-boost, but they don't have the statistics to tell you how protected you are at 9 days post-boost. So from a biological perspective, yes, it's definitely gradual. From a public health perspective, I would say that it's not.
Okay. Why is the J&J one shot, but the others are all two?
I don't know. And fortunately, they don't know either. When these studies are being designed, they have the opportunity, you don't have the opportunity to change it later. You can't give it as a one shot and then decide that 2 is better, or you can't give it as a 2 shot and then decide it works perfectly well after 1 and then market it after 1. The Moderna and Pfizer vaccines are both quite effective after 1, and they take great pains to point that out in the published clinical studies, which is why you might have seen in the news some comments about how the Pfizer vaccine is effective after 1 dose. But they can't sell it that way now, because once a clinical trial is run, that's it. You can't even change how you label the thing afterwards. So why in particular it is probably, I'm not sure that it's actually any different than the others, but they had designed it specifically in that way because they had the luxury to do so, in part because of the size of the organization and where in the vaccine rollout they came. They knew they needed something to improve over others, and they had a very large trial going on at the time. So you're probably reasonably equally well protected after the first dose from all of them, but they specifically set out to test it that way from the beginning.
I think I've covered all the questions. I don't see any raised hands. We are on the dot. One question that Kathy Service tried to answer, and I think she got it correct, is someone wanted your email address. It went too fast on one of the slides.
Certainly,
So
yes.
Marshall Karpel, Marshall with 2 L's at harvard.edu.
Yes. So if you go to the Northampton Neighbors website, you can find the name of this, my name for the talk. My email address is my first name plus my last name. So just Marshall Karpel at G, the letter as in goat, g.harvard.edu.
Okay. Last chance, folks. Unmute yourself and ask a question. This was wonderful. Thank you so much. I think I understand about this much more, but oh, we have a hand.
Question, yeah. I'm sorry if I missed you saying this, Marshall, but by the way, I love your presentation. Thank you. I'm not sure you answered why Pfizer and Moderna decided to test as a 2-shot deal. What made
I'm definitely not qualified to answer, because I assume that decision was made somewhere at the C-suite level of business executives who had market positioning reasons for that. It's certainly not intuitive based on the biological effects that occur. The rationale behind the 1 versus 2 dose is you get the improvement of the antibodies, but you also get better memory retention at the immunological level from a prime and a boost than you do from the prime. Why that particular company chose to test it another way is way over my head.
They were getting a lot of money from the government at the time.
Yes, I suspect it was a business decision and not a
But I
biology decision.
I do remember as a kid getting the first polio vaccine, and that was it wasn't shots. They put a little liquid in a sugar cube and you eat that, but that was a 2 administration deal too.
Yep.
Maybe
And so,
same reason.
and part of the reason for that is because that was an attenuated virus. So it was infecting you and causing a stronger immune response, but because it was attenuated, it didn't infect people very well. And so the immune response was adequate. Giving it as a prime boost allows that secondary exposure whereby the memory B cells really go crazy the second time around and not only respond with greater strength, but also then are sequestered later on for stronger memory.
Thank you.
I had a question.
Go ahead and ask it.
Okay, so I've been in touch with the post-COVID clinic that just opened in Springfield here. My partner and I had mild cases in the spring, and we've got some of the symptoms, not some really severe ones, although he has one that's concerning. But I was told we're gonna be getting referrals there, but I was told that we should not get our second shot. We're still trying to get the vaccine. And that was something that I was told was you won't get the second shot. And I have not heard that anywhere. I mean, I know if it's J&J, it wouldn't be, but we're right now, my partner can't do Moderna. He's allergic to an ingredient in it. And so we've been trying to get Pfizer and staying up in the middle of the night to book appointments at CVS and not getting anywhere. But I'm going to be in touch with them because now I haven't heard that anywhere else. And I'm wondering if that's because of the symptoms or I don't know if you've heard of that or not with post-COVID people.
So I certainly can't comment on anything that specific because I don't know your medical history well enough. I can say that what you're talking about is already off of the, I would say, approved vaccine response profile. You're talking about switching, you had 1 dose of one and now you're talking about
No, we haven't had any yet. We're still waiting. We're still waiting to get one, but they just said they were telling people not to get a second version, a second of whatever one they got, that they didn't need to get the second one.
Got it. For that, I do think it would be inappropriate for me to guess.
I just didn't know if you'd heard about it.
But I haven't heard anything like that. So I would definitely run that by your PCP in addition to the clinic that is reaching out.
Yeah, I'm going to be going through the clinic, but I was just wondering if that was information that other people had been hearing. I hadn't. So anyway, thank you.
Not me. I certainly haven't heard anything like that myself.
Okay, thanks.
Well, Marshall, we had 85 people at one point, or the last time I checked. Some are clocking out now. I'm sure it's going to be watched many times before. Thanks so much for your presentation.
It's my pleasure. Thank you for having me.
Okay. See
Good luck.
everybody.
Stay safe.
Bye-bye.
Thank you.